Hello, everyone. Thank you for joining us for today's webinar. InVivoScribe has been providing products for hemato-oncology research and diagnosis for over 25 years and is the proud sponsor of this webinar. We welcome you to this educational program organized by AMP, the Association for Molecular Pathology. If you would like to ask any questions during today's webinar, please click the chat icon on the left-hand side of your screen and submit your questions. A corporate sponsor will return your email at the earliest convenience. Our program title today is Lab Secrets, Stratification and Prognoses of Leukemia and Multiple Myeloma using Next-Generation Sequencing, NGS. We are very pleased to announce the two accomplished speakers today. The first presentation today is provided by Dr. Ricardo Bombin, graduate of the University of Trieste with specialization diploma in clinical pathology from the University of Udine. He has extensive experience with molecular diagnostics regarding lymphoproliferative diseases. Dr. Bombin's research is focused in the field of lymphomas and leukemia, with particular attention to chronic lymphatic leukemia, an area which he has received several targeted research projects, AIRC and Ministry of Health. The title of Dr. Bombin's talk is The Prognostication of Chronic Lymphocytic Leukemia, CLL, using NGS. Dr. Walter de Brouwer will be presenting his work during the second half of the webinar. In 2017, he obtained his medical degree from the Free University of Brussels and started his clinical residency in internal medicine. After a year of hands-on medical training, he returned to the lab to start his PhD project to evaluate novel methods to assess residual disease in multiple myeloma. Together with Jonah van der Straten, he published an article describing a reliable MRD quantification technique for patients suffering from both acute lymphoblastic leukemia and multiple myeloma in the Journal of Molecular Diagnostics. The title of Dr. de Brouwer's talk is Evaluation of Strategies to Leverage NGS-based MRD in Multiple Myeloma and in Acute Lymphoblastic Leukemia. Dr. Bombin, I will turn the floor over to you.
Okay. Good morning, everybody. And first, let me thank InVivoScribe for asking me to present our experience in the context of prognostication of chronic lymphocytic leukemia using NGS. This will be the outline of my presentation. I will show you the importance of IGHV gene in CLL, the recommendation for the analysis of IGVA gene and how to report the somatic hypermutation. Then in the third part, I try to give you an overview of our personal experience in our lab in the context of the NGS, and in particular in the NGS for the immunoglobulin. And in the last part, I'll try to give you some advice, some tip and advice related to my experience for the NGS. So chronic lymphocytic leukemia is a disease characterized by the accumulation of active B-cell lymphocytes, and it's a chronic, incurable disease, for sure less incurable with the introduction of the new target agent. And it has a highly variable clinical course. In fact, we can distinguish between two main subsets of patients: those who rapidly evolve and require treatment, and those who remain stable for years, they do not require treatment. And for this reason, it's important to have some prognostic factors that we're able to identify these two subsets of patients in order to tailor therapy. Here, I just reported, we have many prognostic factors in CLL. Here, I just reported the molecular prognostic factor. And I want to point the attention in the mutational status of TP53, one of the most important prognostic and predictive factors in the context of CLL. And the other important prognostic factor, I could say the historical one, is the mutational status of the immunoglobulin heavy chain variable genes. In fact, probably you already know that patients that presented a mutated configuration of the immunoglobulin have a significantly better outcome than patients that present an unmutated configuration. And for this reason, the mutational status of IGVA gene is a prognostic factor in the context of chemoimmunotherapy. You can see here an example related to the patient belonging to our core, and it's even a predictive factor since patients that present an unmutated configuration do not benefit from standard chemoimmunotherapy because they eventually relapse. And this is the timeline evolution of CLL therapy. I was just to point the attention on the introduction of the new target agent, molecule against the specific BCR pathway or BCL2 molecule. And with the introduction of this molecule, we have a great improvement in the treatment of CLL. You can see here the difference. You can see here that unmutated patients treated with, the difference between unmutated patients treated with obinutuzumab plus prorambusin respect to the same group of patients treated with ibrutinib plus obinutuzumab. But nevertheless, it seems that unmutated patients are a little bit different even in this context respect to the mutated patient, even in the context of BCR inhibitor or venetoclax therapy. And speaking about venetoclax, you can use the immunoglobulin, the sequence of immunoglobulin, not only for the calculation of the mutational status, but since venetoclax is a therapy aimed at eradicating the disease, you can use the IGVA gene sequence as a tool for MRD. You can use the other PCR, digital droplet PCR, or even the NGS. Since patients that were able to achieve MRD negativity have a significantly better outcome than patients that remain positive. And of course, you can couple the molecular analysis with the flow analysis. And for all these reasons, it's important to analyze, according to all the guidelines for the CLL, to analyze the mutational status before therapy, and according to the ESMO guideline for the management algorithm for patients with CLL, you can use it, the mutational status, even to tailor therapy. Think about a patient with no TP53 aberration, but with a mutated configuration of the immunoglobulin, young and fit patient, you can treat it with a fixed therapy, for example, chemoimmunotherapy. On the contrary, an unmutated patient should be treated with ibrutinib, but of course, depending on where you are and the rule of your country. This slide just to remind that after clonal selection, all CLL cells express the very same immunoglobulin. And here I show you the structure of the immunoglobulin before the recombination, and after the recombination, you have the selection of 1B, 1D, and 1J with more than 10 to 12 possible combinations. And here we come to the second part of my talk, how we could analyze these genes. Here I reported the ERIC recommendation, where they say that you can start from genetic material, both genomic DNA or RNA. You have to perform a PCR using the leader strategy, leader primer plus a JH or a primer in the constant region if you are using the RNA. Then you have to sequence, and then you have to analyze your sequence with the IMGT database to retrieve the somatic hypermutation, and with the RSpool in order to say if a patient belongs or not to a specific subset. Just a very brief overview about IMGT. You simply go to the IMGT request, you put your sequence inside, and this is the result. You have all the information that you need. You have your V, D, J, the different alleles. You have the sequence of the CDR3. You have, and most importantly, you have the number of mutations and the percent of homology. And just to remember that more or equal than 98% of homology, a patient is unmutated, less than 98%, a patient is mutated. And I was asked to say something about the subset identification. And it could happen that different patients in different parts of the world have quasi-identical stereotyped BCR immunoglobulins. But what does it mean? It means that these patients use the same IGVA gene, have at least 50% of amino acid identity and 70% of similarity inside the CDR3, have the same length of the CDR3, and they have an identical offset of the shared amino acid pattern. In principle, it means that they have the same VDJ recombination. And it could happen that patients that belong to specific subsets do not only share the sequence of the immunoglobulin, but often display remarkable, consistent clinical biological profiles. And according to this paper, the last paper about the subset, they analyzed more than 30,000 CLL sequences, and they were able to identify then 12,000 sequences belong to specific subsets. It means that 40% of CLL express stereotype immunoglobulin. That for sure is surprising, thinking about the fact that you have more than 10 to 12 possible VDJ recombinations. And in this paper, they were able to identify more than 5,000 distinct clusters or subsets. But is it important to report all these 5,000 subsets? No, because it's important to report these subsets, subset 1, 2, 4, and 8. And why is it important? Because this subset, for example, subset 1 is associated to a very aggressive behavior, quite expected. All patients are presented an unmutated configuration of the immunoglobulin, and the same happens for the subset 8. Again, an aggressive subset, all patients with an unmutated configuration, the VH39, and it's important to report this subset not only for the aggressive behavior of the patient, but because the patient that presents a sickness that belongs to this subset has higher probability to evolve to Richter syndrome. So it's important to report it in your clinical record. Then we have the subset 4, VH434, a very indolent subset with a very good outcome, quite expected, all mutated patients. And then we have the subset 2, my opinion, one of the most important subsets because it has a very aggressive disease, but patients in this subset were unmutated and mutated. So if in a clinical record you report that the patient is mutated, you can think that the patient could have a good outcome, but since it belongs to subset 2, it has a very aggressive disease. So it's important to speak about the subset. And here you can have an example in the Kaplan-Meier curve. You can see both in the treatment naive and in the refractory patient that the patient belonging to subset 1, 2, and 8 have a very aggressive disease comparable to those of unmutated patients, while patients in subset 4 have a very good outcome. And how you can select or can say that a patient belongs or not to subset? You can simply use these tools that just look for the 19 major subsets, and you can simply put the sequence inside this tool, and here you have your record. Here I just report a patient belonging to subset 2 and a patient do not belonging to any subset. And here I want to report what is an example of our clinical report in our lab. We just not report only mutated or unmutated definition. Here, this is 2 patients analyzed in the context of GenQA quality round. Here, in the report, we usually report the V, the D, the J gene with the different allele, with the percent of homology respect to the closest germline, the definition of mutated or unmutated, a note related to the method that we use, and a comment. For example, patient belongs to mutated category, usually associated with a good clinical outcome. And here you can see that we were able to identify correctly the 2 patients. And coming to the next part, probably the most interesting part, I want just to say a couple of words related to my lab. We are a lab devoted to diagnostic procedures for leukemia lymphoma and devoted to research. And 90% of research is related to CLL and the other 10% related to other leukemia lymphoma. And since, as I said, we are deeply involved in CLL, we analyze CLL since 2002, I can say, and we always analyze the immunoglobulin and other genes. But in 2015, due to the high number of samples, we decided to move all our Sanger sequencing to NGS analysis. And in the context of analysis of immunoglobulin, we decided to try the IGVH somatic hypermutation assay from InVivoScribe, and I say that till now we are using the same assay. And here is just briefly report the flow chart of the kit, starting from genetic materials. You perform an amplification, you purify your PCR product, you perform the quantification, you load on the MySeq in our case, and then you analyze your data with the software from InVivoScribe. And here just a couple of words related to how the software works. The software simply merges read 1 and read 2 in order to create a complete immunoglobulin. This is the case of the Illumina result, then collapse all the sequence in order to create group of sequence with the very same identical sequence, and then aligning to the IG immunoglobulin database. And here you have your report in Excel file. And we simply put the sequence inside the IMGT, inside the RS tool, and we retrieve our result in order to fill in our clinical record. But coming to the NGS part, starting from the end of 2015, we have analyzed more than 2,000 CLL for the immunoglobulin, and only analyzed in the NGS. And for 2,076 samples, we were able to identify a single clonal rearrangement. 65 samples presented a allelic productive rearrangement, and in 62 samples, we didn't see any specific rearrangement. And here I report the use of VH family divided by mutational status, and you can see that the most used VH is the VH3, most of them mutated. Then we have the VH1, most of them unmutated, and the VH4, most of them mutated. And this data is in keeping with the literature data. And here I report just the 12 most expressed IGVH gene in our cohort, and here the same most expressed 12 gene in the cohort related to the 30,000 CLL patients. You can see that we have the same scale, the same percentage of the use of the VH, and we have the same VH. And what does this mean? It means that this kit, our procedure, is able to recapitulate not only the use of VH family, but is able to recapitulate the usage of the different VH gene presented in very wide cohort in the literature. And what about the mutational status? Here I report the data related to patients analyzed only with the NGS for the mutational status of the immunoglobulin, patients treated with standard care immunotherapy, and you can see that patients with a mutated configuration of the immunoglobulin have a significantly better outcome than patients with an unmutated immunoglobulin, both in the context of treatment-free survival and overall survival. And this means that the kit is able even to recapitulate the capacity of the mutational status to predict and to act as a prognostic marker in the context of CLL. Here just personal data, very unpublished, very preliminary, in which we have 300 cases treated with ibrutinib, and we can see that even in the ibrutinib, it seems that the mutational status of immunoglobulin could remain a prognostic factor. And coming to the third part, I want to try to give you some tip and advice. Here, for example, I said at the beginning of my presentation for the ERIC recommendation that you have to use the leader strategy. Of course, for those who are familiar with immunoglobulin, it's quite expected why you have to use the leader strategy, since the blue region is the V gene, and it's the region that you have to use for the calculation of homology and for the somatic hypermutation. And of course, you have to use primer outside this region, the leader primer. And if you use other strategies, for example, the FR1 strategy, primer are inside this region, so you miss, depending on the VH, the first 40, 50 base of this region, and this could lead to two things. One, you could overestimate your somatic hypermutation because the region is smaller, so the denominator is smaller, and you can have some equivocal results for the closer germline identity. Meaning that, for example, for the VH434, the allele 1 and the allele 2 have one nucleotide of difference inside these the first 40 bases. So using the FR1, you could wrongly assign the allele, not the VH, but the allele. But looking to related to the mutational status, there are some papers here where they analyze 50 patients with the leader strategy and Sanger sequencing, and the same 50 patients using an FR1 strategy with the NGS for the PGM, and they found 100% concordance for the unmutated or mutated definition. And here are some our results related to our patient, in which we analyzed more than 500 patients with the leader strategy, and using a bioinformatic trick, we analyze this sequence as if they were generated using the FR strategy. We simply cut the first 30, 50 base of the VH depending on the use of the family. And you can see that for 99% of the sample, we were able to recapitulate the very same result, mutated or unmutated, for the leader and for the FR1. But we have a discrepancy for 7 cases in which 4 of them were unmutated, and you can see here it is quite simple to understand why, because we identify 5 mutations with the FR1 and 5 is a bad number, since 5 is mutated for the FR1 but is unmutated for the leader strategy. And so you have to be very careful if you want to use the FR1 in order to define the mutational status. Usually in our lab, we have this procedure. We start analyzing the leader strategy. Then if we do not have a very straightforward result, we move to the FR1 strategy, and we change material. For example, if we start with the leader and DNA, we move to the FR1 with the cDNA, or vice versa. And of course, we have the capacity to go back to the leader strategy in order to be sure of the mutational status that we report in our clinical lab. And this is a recapitulation. In the NGS, you have another thing with respect to Sanger sequencing. Of course, when you have the Sanger sequencing, you have just at the end of your procedure, you have only just one sequence, so it's quite simple, one sequence that is an average sequence related to the whole subclonal composition of the CLL clone. You simply put this sequence and you have your result. But in the NGS, you have this situation. You have one clone and you have other clones that have the same V, same D, same J, but different from 1, 2, or 3 bases from each other. And you could have a different situation. For example, you can have a situation where you have one super major clone and other very small minor clone. You can have one major clone and a minor secondary clone, and you have 2 clones that they have the same number of sequence. And the first question, is it true or not? Yes, it's true. Because related to our research purposes, we use a leader strategy coupled with the UMI, unipolar UMI, and we perform a specific RT-PCR on the cDNA using a JH primer coupled with the UMI. Then we generate the second strand with the V-leader primer plus the UMI again, and then we generate our library with the index and the adapter for the Illumina. And to cut a long story short, you can see here the result. You have that here you have the major subclone identified using the UMI strategy, and the major subclone identified with the leader strategy with the in vivo scribe assay. And you can see that we have a very straightforward correlation. That means that the major class subclone identified with the UMI have the very same sequence of the major class subclone identified with the leader strategy, and they have the same percentage inside the clone, and the same happens from the minor subclone. And this means that this kit is able to recapitulate not only the general usage of the V gene and the mutational status, but it's even able to recapitulate the subclonal composition of the CLL clone. And then we have another problem. For the Sanger sequencing, you have only one sequence, and you could simply have your somatic hypermutation for that sequence. But here you have many sequences. How you could analyze the somatic hypermutation? First thing to say that there is no rule for the NGS and the analysis of somatic hypermutation. So I will tell you the rule that we have inside our lab. Usually in the case when we have one major clone, we took this clone and we analyzed it in the IEMGT and the RS tool. And then if we have a subclone that accounts for at least 10% of the major cluster of the major subclone, we use the weighted average. To put it simple, if the major subclone is 10,000, 100,000 sequence, we analyze with the weighted average only clones that have at least 10,000 sequence. And to say that my colleague, a bioinformatician guy, that analyze the mutational status of the immunoglobulin using the only the major subclone or all the subclone, and the result was the same. If a patient is mutated, he remains mutated, or vice versa. And here you have one slide. The important thing you can see at the bottom of this slide, two very important reviews related to the use of NGS for the analysis of the immunoglobulin. You have some pros and cons related to the decision to use Sanger sequencing or NGS analysis. And I want to give you my personal opinion. The decision to move from Sanger to the NGS is dependent on how many samples you have. Because if you have a small number of samples and the turnaround time, depending on the time that you need to generate your clinical report, if you have 1 week to generate or 2 weeks to generate your clinical report, but you don't have so many samples, you have to, quote unquote, wait a flow cell. And so it is important to consider the fact that you need to run the flow cell, and you have you need a lot of samples. Usually we run, in our lab, we run a couple of flow cells a week. So we have not only for the immunoglobulin, for the other gene, for the CLL or other lymphoproliferative disease. So we have the possibility to generate mixed libraries. So we have the possibility to obtain the result in just 1 week and to generate our clinical report. And this is my very last slide. I want to thank all the people in my lab, in particular my boss, and Filippo that performed all the bioinformatic analysis, and all our Italian colleagues. And of course, even if I don't see you for your attention, and open to answer to your question by email.
Hi everyone. I'd like to tell you something. First of all, I'd like to thank InVivoScribe for the opportunity to present here today, and I would like to tell you something about the importance of MRD detection in two lymphoid malignancies. The lymphoid malignancies I'm going to tell you about are, wait a moment, yeah, perfect. So, are acute lymphoblastic leukemia and multiple myeloma. And why have we focused on these two malignancies? Well, acute lymphoblastic leukemia is the most common malignancy in children, and it is characterized by clonal expansion of the B and T cell progenitor cells. Most children with the therapies available today have a good prognosis, and we can cure almost 90% of those children. In multiple myeloma, however, there is no cure yet available. The novel therapies succeed in attaining high remission rates, but almost all patients relapse with the residual disease that is still present. It is the second most common hematological malignancy in those adult patients and is characterized by clonal expansion of terminally differentiated B cells, the so-called plasma cells. Why is MRD detection important in those two populations? In multiple myeloma, it is of course important to detect whether there is a lot of residual disease that cannot be picked up by the conventional remission criteria, or if there is an absence of MRD with the most sensitive techniques today, as this has been associated with better prognosis. In ALL, on the other hand, most patients are cured, but most patients are also very young. So we want to avoid overtreatment of those patients who are already cured, and at the same time minimize the risk of relapse for those patients who need more intensive therapies. And some studies have been done. So I'll just take my pointer. And then in this study in ALL, they've designed a trial where they decreased the intensity of the chemotherapy used in those patients who were MRD negative, and found that they had an exactly identical survival curve compared to the patients receiving a standard regimen. So we could safely decrease the chemotherapy without increasing the risk of relapse in those patients, and thus minimizing toxicity. In multiple myeloma, these curves show that there is significant correlation between an MRD negative status and prognosis over time. In the next slide, we see that in the ALL population again, we see that when patients are MRD negative, though MRD positive, and we intensify treatment, they have a better prognosis than those patients who receive the standard of care. So if the patients are MRD negative, we can safely decrease therapy to avoid toxicity. And if the patients remain MRD positive, we should increase therapy to avoid that they relapse later in their disease course. In myeloma, similar projects are going on, and these data were presented at the latest iteration of ASH, where Dr. Costa presented results from the MASTER trial, showing that therapy could be safely reduced in those patients who received an MRD negative result in 2 subsequent analyses. Why is a lymphoid malignancy such an eager target for MRD analysis? Well, as Dr. Bomben described before, it contains a unique gene sequence, which is necessary for vast variability of the immune system. We need a diverse gene repertoire. And this is managed by recombination in the lymphocytes. So every leukemic clone or every myeloma cell contains the same clonal sequence, which is only found in these cells and none of the other cells in a body, creating a molecular fingerprint which we can track over time. In a normal situation, there is an abundance of different lymphocytes which all contain unique sequences with rearranged VDJ sequences. However, in myeloma or ALL, there is a malignant population which constitutes an important part of all VDJ rearranged sequences, in this case, about 50%. And then by next generation sequencing, we can characterize this sequence and we can start treatment. After treatment, if we are 100% successful and the disease is gone, then we see that we return to a normal situation. However, in myeloma, much more frequently, we find that not every single myeloma cell is gone. And we can see here that there is a residual clonal sequence still present hidden beyond the other sequences, but this can be detected with next generation sequencing. So how do we go about to detect the clonal sequence? When we have a diagnostic sample, we extract the DNA, and in single-step PCR reaction, we amplify the immunoglobulin or T-cell receptor sequences, while at the same time joining them with adapter genes necessary for the PCR reaction. We do this for several different genes. And in the prior times, we did this for ALL for as many loci as possible, both immunoglobulin loci as well as T-cell receptor gene loci. And there was a slight difference between B and T-cell ALL in the loci we examined. For myeloma, as it was a B-cell malignancy, it was only done for the immunoglobulin sequences. After the amplification, we have to purify the amplicons by a magnetic bead purification step, after which we characterize the length of the sequences and the amount of DNA we are presented with. And this can be then, this can progress to the next generation sequencing device, the MySeq. After 39 hours, we get the files which tell us which sequences are present in the sample. And as you can see in this sample, we have we have one sequence which is abundantly present in this sample. And if the sequence is present in more than 5% of the reads, then we can call this a clonal sequence and use this for follow-up in an MRD setting. An MRD setting, we can choose for either next generation sequencing in the same steps as I just described, or we can choose to design patient-specific primer and probe combinations and continue with quantitative ASO-PCR, which has been the reference method up until now. However, the design of primer and probes, which are patient-specific, is laborious and requires quite some expertise. The primers and probes are designed against the junctional and most variable region of the immunoglobulin or T cell receptor. And then after optimization steps, which takes about, all in all, it takes about 3 to 4 weeks, after which, if we are successful, we can continue with a ASO-PCR design in which the fluorochrome is released if the clonal sequence is present, which we can then detect and so estimate the presence of clonal sequences in a sample. However, it does not allow a direct evaluation of the sequence, and so there is a remaining risk of false positive results. But if the design of the ASO primer was successful, it allows for a cheaper and faster evaluation of the residual disease compared to next generation sequencing, which is internationally standardized by the Euro-MRD panel. Some negative sides of the quantitative ASO-PCR are the limited number of sequences that can be monitored, as we need to design patient-specific primer and probe combinations. For every patient, a maximum of 2 primer-probe combinations is made available. And moreover, we need diagnostic DNA for the design of a standard curve to quantify the amount of tumor DNA still present in the sample. Moreover, in myeloma, where we have hypermutated immunoglobulin sequences, ASO primer design is more difficult and only applicable in 50 to 70% of the patients. Those problems are less pronounced with next generation sequencing, where we can just sequence all reads and all sequences that were clonal at diagnosis are followed up at the follow-up samples. Moreover, we do not need to design a standard curve. We can quantify the residual disease just by our reference molecules, as I will explain to you later on. Moreover, it allows a direct evaluation of the clonal sequence, so there is a higher specificity. There's no risk of positivity due to highly similar sequences, as we can directly compare the sequences to one another. And in myeloma, it has a higher applicability compared to the quantitative ASO-PCR. Next generation sequencing is available for 90 to 95% of the patients instead of 50 to 70% of the patients. Moreover, we do not have to design patient-specific primers, which often fails and thus requires a lot of loci to be interrogated to find 2 primers which might work. We can just continue with the sequences already detected at the first diagnostic NGS run. But every NGS run is more expensive than an individual quantitative ASO-PCR run and is more time consuming. Once ASO-PCR is done and the primers are defined, it can be done in about a day, while NGS takes about a week before we can get the results. Moreover, next generation sequencing is not yet internationally standardized, where ASO-PCR is standardized. For the detection of the clonality, we compared, we evaluated 170 samples, 180 samples in total, and found that the immunoglobulin heavy chain, light chain, and T cell receptor gamma chain primers from InVivoScribe succeeded to detect 93% of all in this clonal sequence in 93% of the patients. In some patients where we could not detect a clonal sequence with one of these 3 primers, we could continue with TCR beta and TCR gamma, TCR delta primers, and found that in an additional 5% of the patients, which is very limited, we could detect an additional clonal sequence. So these these primers from in vivo scribe seem to be quite effective for the detection of a clonal sequence in these samples. In an independent myeloma cohort of 14 patients, we evaluated next generation sequencing and compared it to Sanger sequencing, and found that for all of the 14 patients, we found at least one clonal sequence, while for the Sanger sequencing in one patient, we could not detect a clonal sequence. This is a minor difference, but additionally to this, next generation sequencing could also detect additional clonal sequences in 10 out of 14 patients, suggesting that next generation sequencing is much better at detecting clonal sequences at diagnosis than the conventional Sanger sequencing is. So at diagnosis, and next generation sequencing seems superior, but how do we quantify minimal residual disease? Well, after extraction of DNA from the lymphocyte population, we can quantify the number of total cells by albumin PCR, and then we add some reference molecules to the mix, after which we amplify in the PCR step I described previously all DNA samples, and then we sequence these amplicants. After correction for the quantification of the reference molecules, the known amount of reference molecules, we can then correct the percentage of leukemic reads by the quantification rate of the reference molecules, and so determine exact residual disease, amount of residual disease. And this is what we tested in a next dilution experiment, where we diluted known tumor quantities in different concentrations and found an excellent correlation between the expected MRD as we diluted it in the samples and the MRD we could detect by the next generation sequencing method. One sample, however, at 5 times 10 to the minus fifth was negative by next generation sequencing method, but this was probably due to a lower DNA input, which impedes optimal quantification at those low concentrations and can be overcome by adding more DNA to the next generation sequencing run. For the accuracy and to determine whether next generation sequencing was as good as the gold standard quantitative ASO-PCR, we evaluated 59 samples and detected MRD by both quantitative ASO-PCR and next generation sequencing. And we found that in 29 of the samples, both analyses were positive, while in 11 samples, sorry, I always say percentages, but in 11 samples, they were both negative. In 13 samples, we had a positive next generation sequencing result, while the quantitative ASO-PCR was either low positive or negative. In 12 of these 13 patients, we could detect by next generation sequencing the clonal sequence at a level below the quantitative range of the ASO-PCR method. So an additional 22% of the samples could be quantified by next generation sequencing if we compare it to the gold standard quantitative ASO-PCR, suggesting a higher sensitivity and specificity of the next generation sequencing approach. On the other hand, there were 6 samples which were negative with next generation sequencing, but low positive with quantitative ASO-PCR. And there could be several explanations for this, one of it being a low DNA input. So we reran every sample and found that in 3 of these samples, we had a positive result at very low concentrations in the rerun sample. So at these low concentrations, we are really at the limit of detection of the next generation sequencing method for those DNA inputs, and we could expect a negative value. So it is important for MRD detection to measure your samples twice. For 3 of these samples, however, we could not detect a positive sample We could not detect any residual disease even in the second run. This suggests that it's either a false positive quantitative ASO-PCR result or a false negative next-generation sequencing result due to low DNA input. Here in this slide, I retake what I've just said, and so there are two possible explanations for this. We tried already setting a second experiment for next-generation sequencing, and it didn't work to detect the clonal sequence. One other possibility is use a higher DNA input to detect the clonal sequence. And this has been tried by us, which I will explain in two slides. First of all, I'll say something more about the false positive ASO-PCR results. As was shown in 2017, in patients with ALL after transplant, sometimes the quantitative ASO-PCR result was low positive while the patients remained in remission. And if they went on to analyze the sequences by next-generation sequencing, they found that there were similar sequences which could make for an aspecific primer binding of the quantitative ASO-PCR when primers and probes bound to highly similar sequences but not identical sequences, thus resulting in a false positive result, while next-generation sequencing would clearly take these two samples as being completely different. And as I promised you, we also tried to do a dilution experiment in which we try to put as much DNA in one next generation sequencing run to increase the sensitivity. And in this case, we diluted some tumor cells in the DNA of healthy donors at different concentrations, just about one tumor cell in a million normal cells. After which we concentrated the DNA through vacuum evaporation to a concentration of 7 micrograms DNA input in one next generation sequencing run. We asked for 1 million reads and found that we could actually find a perfect correlation between the theoretical input of MRD and the next generation sequencing results of MRD without any inhibition that was due to the vacuum evaporation method. This signals that we can increase the DNA input in a single and next generation run and thus increase the sensitivity of next generation sequencing. However, for myeloma, next generation sequencing has added results. As I told you before, in 14 patients, quantitative ASO-PCR was only available for 57% of these patients, 8 out of 14 patients, while next generation sequencing allowed MRD quantification in all patients. So additionally to the patients we could follow up by quantitative ASO-PCR, we could follow 4 out of 10 patients extra. And when we compared the ASO-PCR to the next generation sequencing method, for those patients in which an ASO-PCR was available, we found a good correlation between the next generation sequencing method and the ASO-PCR, as well as some samples that were again positive with next generation sequencing and negative or low positive with the ASO-PCR method, suggesting that an additional 36% of the samples could be quantified by next generation sequencing, which means that it has a higher sensitivity and specificity with this approach than the current ASO-PCR approach. We found no samples that were positive by quantitative ASO-PCR and negative with next generation sequencing. So in conclusion, I would say that next generation sequencing by the 3 primers provided to us by InVivoScribe succeeded to detect 93% of all VDG rearrangements, clonal rearrangements in ALL and multiple myeloma. Moreover, the assessment of MRD by next generation sequencing was accurate with a good correlation with the gold standard quantitative ASO-PCR. The only disadvantage of the next generation sequencing method at this moment is the longer turnaround time. The results I just described were all published in a recent article in the Journal of Molecular Diagnostics. How does this translate to our current lab setting in clinical practice? Well, we use next generation sequencing for target detection in ALL and diagnostic samples. After this target detection, we design ASO primers and continue follow-up with the quantitative ASO-PCR for the moment, as international standardization is still lacking. CLL is for CLL, we also use the next generation sequencing approach to calculate the somatic hypermutation rate in these patients. And in MRD, assessment in myeloma is only done at the request of the demanding physician and in research settings. But in ALL, it is done when there is not enough diagnostic sample available for a standard curve, so no ASO-PCR can be performed, or when there are no good primer-probe combinations for ASO-PCR, or if we find a low positive ASO-PCR result, which is unexpected or found in a post-transplant setting, then we want to verify these results with the next generation sequencing method. In my last slide, I would just like to thank all the funding agencies, in VivoScribe for the opportunity, and my promoters for helping me do this research, Professor Van Riet, Professor Bacchus, Professor Schots, and all the staff of the Lab of Molecular Hematology. I would like to thank you all for your attention, and I'm looking forward to answer your questions by mail later on.
On behalf of VivoScribe and AMP, we want to thank everyone for your attendance today at this webinar. Please note that a recording of this webinar will be sent to all registrants, and any questions will be addressed in a follow-up email. We would especially like to thank Dr. Bombin and Dr. DeBrouwer for their excellent presentations and for sharing their experiences with us today. Thank you all so much for your attendance, and have a wonderful afternoon.